Good
Morning, ma'am.
morning. Good morning. Good morning.
Good morning, ma'am.
Good morning.
Good morning, ma'am.
Good morning.
Ma'am,
Yes.
ma'am, are columnar cells multinucleated?
Who said that?
In the picture it was given, ma'am.
Watch carefully. Watch carefully, Nani. That's NCRT picture. One nucleus is up, one is below. Watch carefully.
Yes, ma'am. So is it not multinucleated?
No. So we are learning about simple epithelium. Simple epithelium. Epithelium is 3 types based on shape of cells. So one single layer of cells stand on a basement membrane. Then if the cells are very flat, like tiles on a floor, like this. Sometimes this is the surface view. This is the side view. Thickness you see. These are very thin, flat cells looking like tiles on a floor. This is the surface view, right? This is the thickness. This is the thickness. Such flat cells, which are flattened nucleus, represents squamous epithelium. So the cells are very much flat. This is the surface view. From upper surface, this is the surface view. If you look at it, it looks like one layer of cells standing on the basement membrane, and the cells are very thin. This is the thinnest epithelium. This is the thinnest epithelium. No other epithelia is thinner than this. This is the thinnest epithelium. Then it is also called pavement epithelium because substances walk across this epithelium. Pavement epithelium. It is pavement epithelium because the cells look like tiles on a floor. They are very thin. You might have seen flooring tiles. Very flat, thin. And substances can walk across easily because thickness is very. This type of simple squamous epithelium, where do you find? You have to remember how the cells are and the location, very important. In our body, where these are. Now, you know our lungs have bunches of grapes-like structures. Any idea?
It's
Alveoli, like this. They look like bunches of grapes. These are the air sacs, bags-like structures. And here, oxygen comes into blood, carbon dioxide is given out. So, there is a diffusion of gases. Diffusion of gases across this membrane. So, it is the thinnest epithelium. If thickness is less, gases can move faster. So, diffusion is allowed very quickly here because this is the thinnest layer. So, one example for location of simple squamous is alveoli. Then, so at the alveoli, gaseous exchange is facilitated by simple squamous epithelium. Then, this simple squamous epithelium is seen in the inner surface of heart, blood vessels, arteries, veins, capillaries they have. Lymph vessels, so many locations. Now, lymph is a colorless fluid. I hope you know. Blood is red colored with RBCs. Lymph has WBC but no RBCs. So lymph is a colorless fluid. Wherever this blood or lymph is flowing, the inner surface layers, suppose this is a blood vessel and this is the wall of the blood vessel. Right? This is the wall of the blood vessel. And blood flows here. Like that, lymph flows in lymph vessels. Next. The innermost layer of the blood vessel. Innermost layer of the blood vessel, which is in contact with the blood. And in the heart also, blood flows. This is the heart. These are the 4 chambers. And here is the blood. And this is the wall of the heart. In this wall, the inner surface, which is in contact with the blood, is called endothelium. No doubt it is epithelium. If epithelium envelops blood, note down meaning. Epithelium envelops blood, then it goes with the name endothelium. So epithelium enveloping blood is known as endothelium. This is also made of, come on.
Simple squamous.
So we are getting many locations. I'm just giving example and meanings where they are. You have to remember locations. Alveoli, lung, air sacs or bulbs-like structures where exchange of gases occurs. Endothelium, you may not know the meaning. Note down the layer of epithelium which envelops or surrounds blood is called endothelium. Okay, so epithelium forming the inner surface of lymph vessels, blood vessels, heart is, come on, endothelium. Endothelium. Then that's the meaning. Epithelium, which is enveloping blood and lymph spaces, is called endothelium. If you take capillaries, The capillaries have squamous epithelium. What is this layer called?
Endothelium.
Endothelium.
If you take arteries, veins, their wall is thick. They are large vessels. Their wall is thick. They have many types of cells in it. Not only epithelium, connective tissue will be there, muscle will be there. But is a capillaries. If they are to be taken, which envelopes blood and lymph. Of course, it is in the inner surface of the heart also. Then, can you identify this part in our body?
Bowman's
Bowman's
capsule.
capsule.
Good. Bowman's capsule. Where do you find Bowman's capsule?
Nephron.
In the nephron. And what are these capillaries?
Glomerulus.
Glomerulus.
Nephrons are in our kidney. Our kidney has 1 million nephrons, each kidney. Now these are glomerular capillaries. What's the use of our nephrons?
They have to produce
Filtration
Of kidneys.
Filtration of blood.
Yeah.
filtration.
Yes. They filter metabolic waste such as urea and produce urine. So nephrons make urine. They remove nitrogen waste from our body, excess salts, water, nitrogen waste. Now, if they are to be filtered here, it should be thin or thick.
Thin.
Thin, ma'am.
This is a capillary. Capillary has what? Endothelium.
Endothelium.
Sometimes squamous epithelium may have irregular boundaries like this. Boundaries. Now, here the squamous epithelium has certain leg-like projections like this. These are called podocytes, modified squamous cells. So, don't worry, you will get this again in kidneys of human. Now you remember Bowman's capsule, glomerular capillaries, or any capillary is lined by endothelium. Then Bowman's capsule wall also has, come on, simple squamous epithelium. This allows quick filtration of metabolic waste. So watch, in the alveoli it is allowing quick diffusion of gases. It's a thin lining which can facilitate quick or fast diffusion, and at the blood vessels it can allow fast filtration of nutrients as well as diffusion of gases to tissues. Followed? So what is endothelium?
Yes, ma'am. Epithelium lining the blood, envelope the blood.
Lining or surrounding the blood.
Then I told you about peritoneum. What is peritoneum?
Is the layer covering the coelom.
If you cut any animal with a true coelom, this is the inner tube. This is the outer tube. Tube within tube organization. One tube inside another tube. Gottamlo gottam. The inner tube is the gut, alimentary canal. Outer one is the body wall with the skin and muscles. This cavity is coelom. And this coelom is also lined by
Epithelium on both sides.
Yeah, epithelium. These two layers, coelomic epithelia, are known as peritoneum. Even that has coelomic epithelium. Then what is the peritoneum covering lungs?
Pleura.
Very good. Very good. Pleura. It also has simple squamous epithelium. So whatever cavities, coelomic cavities are there, they are lined by this peritoneum. Parietal layer, visceral layer. The outer layer is parietal layer. And this layer covering the gut or intestine is visceral layer. Then, if it surrounds heart,
Pericardium.
Yeah. Peri means surrounding, cardia meaning heart. Who is a cardiologist?
Doctor who treats the heart.
Yeah, heart specialist. Now, The layers which cover the visceral organs, are also called mesothelium. In NCERT, you will not get this word in this chapter. In human digestive system, he says, this layer as mesothelium. So note down now only. So if it encloses, that is peritoneum, enclosing visceral organs, is called mesothelium. What is endothelium?
Epithelium covering the blood.
Epithelium and
So these are technical terms. You need to know the meanings. Okay. Then the heart has pericardium. Actually, this pericardium is two types. Let's say this is the heart. And there is a covering around the heart. For your understanding, I am drawing a gap. This is the covering. In this heart covering called pericardium, there are two parts: outer pericardium, inner pericardium. Outer one is with collagen fibers. It is fibrous. Most abundant protein, I think in botany it was also mentioned in proteins. Most abundant protein in animal cells is collagen. So this outer layer is a tough covering, fibrous layer. And the inner part of it is called serous pericardium. Seriously learn. Sera meaning watery fluid. So this pericardium is nothing but peritoneum, which has two layers, visceral layer, parietal layer. Between them, there is a watery fluid, pericardial fluid, similar to coelomic fluid. It is similar to coelomic fluid. Okay? So, you get this word, serous pericardium, in your material. Example. NCERT may be giving only 3 examples: alveoli, endothelium, and peritoneal cavities. Okay? So the cavities, coelomic cavities, are enveloped by this peritoneum, and if it covers the peritoneum covering the viscera, meaning of viscera?
Internal organs.
Internal organs.
That's about squamous epithelium. Okay? Simple squamous epithelium. Now let us recall points. How are the cells in simple squamous epithelium?
Flat pavement line.
Flat, tiles-like, the thinnest cells, right? And pavement epithelium, and the nucleus is in the center, somewhat compressed, flattened. Large nucleus only, but flattened, not ball-like. Then, this facilitates easy filtration and diffusion, and it is seen locations. Let's recall: alveoli,
endothelium, mesothelium,
Mesothelium.
blood vessels,
mesothelium,
mesothelium,
Bowman's capsule.
Viscera.
Mesothelium is covering the cavities, right? Peritoneum refers to coelomic epithelium. Then it is seen in the Bowman's capsule also. These are the locations. And in the heart, it forms serous pericardium, which is a outer covering layer with a fluid for shock absorption. Though you jump, skip, run, there won't be shock to your heart because the fluid in the serous pericardium is a shock absorber, lubricant. And even around lungs, you find pleura. Even it has pleural fluid in it. So these are the locations. Everybody, is it clearly understood?
Yes, ma'am.
Yes, ma'am.
Everyone respond.
Yes,
Is
ma'am.
it clearly understood?
Yes. Yes.
Then shall I continue with the next epithelium? Simple cuboidal epithelium. Here the cells are cube-like. Cuboidal means cube-like with a large spherical central nucleus. So a single layer of cuboidal cells will be seen on the basement membrane. So simple cuboidal epithelium has a single layer of cube-like cells or cuboidal cells. Eidos means form. Now, sometimes the surface, this is the free surface facing this is the basement membrane. Below basement membrane, what tissue will be there?
Connective tissue.
Cell lamin.
Good. So below the basement membrane, you will find connective tissue with blood vessels, which is useful for nourishing this. So this is the free surface. On this free surface, sometimes cuboidal epithelium may have cell membrane folds. Important small cell membrane folds. So suppose this is the cell. This is the cell membrane. Cell membrane folds like this, forming like a brush, right? It looks like a turkey towel. Turkey towel, you know?
Yes, ma'am. Yes, ma'am.
Brush like. This is the brush border. These are called microvilli. Don't say villi. Villi are finger-like folds of small intestine. I'll come to that point shortly, don't worry. Villi are finger-like folds of small intestine wall. Microvilli, cell is microscopic. The cell membrane folds on the surface are called microvilli. Can anybody guess the use of this?
Create lash.
Increasing of surface area.
Increase surface area.
Yeah, they increase surface area. Now, where you find cuboidal epithelium, note down, cuboidal epithelium with microvilli is seen in nephrons. It's a seen in nephrons. And the parts of nephron. This is the Bowman's capsule. Any idea about this coiled part?
Proximal convoluted tube.
Proximal convoluted tube.
This is
Loop of
loop
Henle.
of
Loop of
Henle.
Henle.
U-shaped structure is loop of Henle. This is Bowman's capsule. Then again it is folded like this.
Distal
This
convoluted.
is called
Convoluted tubule.
distal convoluted tubule. Convoluted means folded.
Folded.
Tubule means microscopic tube. Distal meaning far. Distal meaning far. Proximal means near.
Near.
So this part is nearer to Bowman's capsule. Shortcut PCT. Here DCT. Okay. In this PCT, you find this brush border. Now when blood gets filtered, glucose, amino acids, vitamins, because of their small size, they get filtered. Can we lose those in urine?
Ma'am.
No. We can't lose glucose, amino acids, certain salts like sodium, etc. through urine. So here filtration is finest filtration because it is simple squamous epithelium. Whatever small molecules are there, they get filtered. They have to be taken back. They have to be taken back. So this cuboidal epithelium, the surface facing this cavity has a brush border. It has a brush border. So if this is the proximal convoluted tubule, these are the cuboidal cells. This is the basement membrane. This is the free surface through which urine is flowing. Here you find this brush border. Okay? The cell membrane folds will be towards this. So these will absorb, come on, nearly 70% of the filtered urine back. Water is taken back. Salts are taken back. Glucose, amino acids, vitamins are taken back. So, to increase surface area of absorption, in the proximal convoluted tubule, cuboidal epithelium has a brush border. So, very important bit. In which part of human body you find cuboidal epithelium with the brush border?
Nephrons.
Now, more clear, in the
CT and
nephron,
DC.
Bowman's capsule is not having, loop of Henle is not having that. You must clearly say PCT. Your options, PCT, DCT, Bowman's capsule, loop of Henle. What do you have to choose? PCT.
CT.
Then, in the distal convoluted tubule, there is cuboidal epithelium. From what I did, can you say any point? In the DCT,
Microvilli.
I said it is there.
Border.
Yeah, brush
Brush
border
border.
is absent.
Absent.
Microvilli are found in the PCT.
PCT.
In the DCT, simple cuboidal epithelium, but without brush border. So, it can eliminate certain waste and it can reabsorb. Secretion, absorption here. Secretion is a process where some waste can enter into this. In the kidneys, that secretion meaning. Don't take it digestive gland or saliva secretion. Okay. So one location, simple cuboidal epithelium with brush border, PCT. Simple cuboidal epithelium without brush border, distal convoluted tubule. Then, have you heard bronchioles?
Yes, ma'am.
Yes, ma'am.
Yes,
Bronchioles. This is our trachea.
ma'am.
Trachea divides into bronchi.
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 Bronchi divide and branch into fine structures.
Bronchioles.
These finest branches are called bronchioles. Here, cuboidal epithelium with cilia. In botany, cell biology is completed. Am I right?
Yes, ma'am.
Cilia flagella structure was taught to you?
Yes, ma'am.
Yes, ma'am.
Then, these cilia are motile. Motile meaning?
They can move.
Yeah.
Action movement.
So, there will be cilia here in the respiratory tract. Can anybody guess the use?
Ma'am, roaming.
Help in
Remember.
pushing out the expired air.
Any dust particles come inside with the lashing movements, they throw them out.
Good. Correct. So, dust particles, microbes enter through air into our respiratory tract. We need to inhale. So, at that time, air containing microbes, dust particles enters. Then, there will be a sticky substance called mucus. So, these are all kept moist. There will be a sticky substance here lining this. Sometimes when you get throat infection, you get lot of phlegm from your throat. Yes. When you cough, thick fluid comes. Now, the cilia here push that mucus. It will trap antigens of the bacteria and microbes, and it will trap dust particles. If the cilia do not push it out, what happens? Your lungs get filled with water, this fluid. Then you can't breathe. So, this mucus is regularly secreted. So, it has to take away the cilia, push that mucus containing dust particles and trapped microbes. That's a use. So, bronchioles have cuboidal epithelium with cilia. Microvilli are cell membrane folds. Cell membrane folds, microvilli. Cilia are having, cilia are having 9+2 arrangement. They arise from a basal granule. All this was taught in botany. Hair-like structures, permanent structures. They are motile. These are to increase area of absorption. Microvilli is to increase area of absorption. Then, in the ovary, this is the ovary of human. It is covered by an epithelium with cuboidal cells. This epithelium covering the ovary is called germinal epithelium. Germinal epithelium. So we completed simple cuboidal epithelium. If you want, you can take one more example. In the thyroid gland, inside, if you cut it, you find follicles like this. These secrete thyroxine hormone. These are called thyroid follicles or vesicles. A follicle is a ball of cells that you will come across in thyroid gland later when you study about human. At that time, you must recollect these examples. Which epithelium in which part of human body. When you go to kidney, you must say Bowman's capsule has squamous epithelium. PCT has cuboidal epithelium with brush border. DCT, that is distal convoluted tubule, has
Cuboidal epithelium without brush border.
The microbiome.
Very good. Very good. Shall we proceed to simple columnar epithelium?
Yes.
Yes, ma'am.
Okay. Simple columnar epithelium. Simple columnar epithelium. Here, the cells are tall, cylindrical, pillars-like, elongated cells. So, one layer of cells standing on the basement membrane. One layer of cells standing on the basement membrane is simple epithelium. Now, here cells are tall, cylindrical, slender means narrow cells, elongated or pillar-like. Then, their nucleus is oval. It's different from other epithelium. It's oval, not in the center, closer to basement membrane. What is the secretory cell organelle in the body? Cell body. Cell. Which plays a good Golgi complex. So, there is a large oval. For cuboidal epithelium, spherical. Large nucleus in the center. For cuboidal epithelium, large
Spherical
ball like
center.
nucleus. And here it is towards
the basement.
Come on,
Base.
basement membrane.
Oval.
And here Golgi complex is well developed. It can produce mucus. So, usually in secretory areas where mucus is secreted, you find this type of epithelium. So, there are special cells that secrete mucus.
Blood cells.
Good. Mucus is a sticky fluid which can trap microbes. So, the special cells that secrete mucus, they are known as goblet cells. They are known as goblet cells. In our respiratory tract, goblet cells are there. In our digestive system, intestine, goblet cells are there. This type of epithelium is seen in the mucosa of stomach and intestine. Note down meaning. Mucosa is a mucus secreting membrane of stomach and intestine. This is what? Stomach. And this one is intestine. This is the cavity. This is the wall. And inside this layer is mucosa. So this is a cavity. Any free surface is enveloped by, come on.
Yeah.
Here is epithelium. That is called mesothelium, covering layer. This is the outer covering, visceral layer or coelomic epithelium. The inner cavity is lined with this columnar epithelium. There, gastric glands secrete gastric juices. So you find simple cuboidal epithelium in mucosa. Correction needed.
Columnar.
You need to respond అవ్వాలి. You must shout loudly. No, columnar. Are you in trance? Simple columnar epithelium lining the cavity of stomach and intestine is called mucosa. Simple columnar epithelium lining the cavity of stomach and intestine is called mucosa. In the stomach, it is simple columnar epithelium. Right? You call it gastric mucosa. What is the word gastric refers to?
Stomach.
Yeah. Then, in the case of small intestine, not large intestine, this type of epithelium will be there. In the small intestine, the mucosa is thrown into folds. The mucosa is the layer. It is thrown into folds. This entire finger-like structure is called villi. Villus is singular. Okay. Now, this is what? Columnar epithelium. Now, the folds of mucosa will, and this mucosa contains simple columnar epithelium, and the free surface has a brush border of microvilli. This is a microvilli. Then gastric mucosa, epithelium.
Columnar epithelium.
Columnar epithelium.
Cuboidal epithelium with
Microvilli.
Border.
Fresh border.
Microvilli. Then cuboidal epithelium with cilia.
In the
Small bronchioles.
bronchioles. Bronchioles.
Bronchioles. Then gastric mucosa.
Columnar epithelium.
Columnar epithelium.
Without breast
Simple
border.
columnar epithelium.
Columnar.
Okay. Let me write completely, otherwise you think C for cuboid. Columnar. Any brush border here? No.
No, ma'am.
In the small intestine, not large intestine, the same epithelium, simple columnar epithelium is with the brush
Microvilli.
Fresh border.
border, that
Microvilli.
is
Microvilli.
microvilli. Okay? Then sometimes this epithelium shows, columnar epithelium shows special cells that secrete mucus. Mucus is a sticky fluid which traps dust or microbes. Okay? What they secrete?
Mucus.
Mucus.
Then sometimes the surface of the epithelium, if it shows cell membrane folds which are very small and perform absorption, in the small intestine absorb digested food, these are called microvilli. Then one question. Does gastric mucosa show microvilli?
No, ma'am.
No, ma'am.
No, ma'am.
Small intestine mucosa,
Yes, ma'am.
it shows.
Yes.
So this is in small intestine mucosa. So the free surface may show cell membrane folds which absorb. They are called microvilli. And in the gastric mucosa, no microvilli and no cilia also. Then in the bronchioles, come on,
Cuboidal
Cuboidal epithelium with cilia.
cuboidal
Cilia.
cells with cilia. In the PCT, cuboidal cells with microvilli.
With
Breast.
microvilli.
Microvilli.
Okay? So be careful while attempting answers. Then sometimes the cuboidal cells or columnar cells may have cilia. Either cuboidal cells or columnar cells may have cilia. Where do you find cuboidal cells with cilia?
Bronchioles.
Bronchioles.
Brush border cuboidal epithelium.
PCT.
PCT.
Proximal convoluted tubule of nephron. Sometimes the columnar epithelium may show cilia, which are motile. So then you call it ciliated. So if a cuboidal cell has cilia, location can be bronchiole. And here, columnar with cilia.
Bronchi.
Do you know fallopian tube?
Fallopian
Don't say ovary. Ma. Ovary doesn't show cilia. Yeah. Fallopian tube is meant for pushing pushing
Egg
the ovum.
towards the uterus.
Egg, uterus.
You have learnt about human reproduction, at least little. So, this tube, which is meant for a transport of ovum, is the fallopian tube, oviduct. Right? It collects the ovum and the cilia in it, push it towards uterus.
Uterus.
So, note down. The cilia in the fallopian tube help in pushing the ovum or zygote towards the uterus. Then it can be seen in, this is hollow. Is it hollow? Tube is hollow inside.
Yes, ma'am.
Yes,
Then
ma'am.
bronchioles are hollow.
Hollow.
In bronchioles it is
Cuboidal epithelium.
cuboidal cells with cilia.
Cilia.
Okay. Next, in our brain and spinal cord, there is a fluid inside, flowing inside.
Cerebrospinal fluid.
Cerebrospinal fluid.
Yes, sir.
So brain has cavities with cerebrospinal fluid. These cavities are called ventricles. ఎక్కడో వచ్చిందే ventricle name?
Heart.
Heart, ma'am.
Heart chambers. So you must say ventricles of brain. There you find ciliated epithelium. Can you guess the use?
Helps to move the cerebrospinal fluid.
Good. They help to move the cerebrospinal fluid. And spinal cord has, even spinal cord has a cavity with the same fluid that is called central canal.
Canal.
Right? That is called central canal. So, in the central nervous system, that is brain spinal cord, these two represent central nervous system. They have cavities in which a fluid is there, cerebrospinal fluid. Okay? Sometimes the epithelial cells have cilia which are motile. They show 9+2 arrangement. Cilia are short hair-like structures which can move. Then cell membrane folds.
Microvilli.
Microvilli.
Yeah. Then in our inner ear, you may find with very long cell membrane folds. They look like cilia. They are not showing 9+2 arrangement. This is seen in our inner ear. These are hair cells. These are not motile. They are not so small like a microvilli. They are cell membrane folds, but they are not for absorption. They are for some mechanical. They are not motile. नाम के वास्ते cilia, not काम के वास्ते. Means they are not motile. They don't show 9+2 arrangement. It's a cell membrane fold which is very long. Because of this long nature, it may look like a cilia. They are called hair cells. They are seen in our inner ear. So, the surface of an epithelium may show cell membrane folds which help in absorption, microvilli. It can show motile structures called cilia, and it can show long, very long cell membrane folds that may look like cilia, not showing 9+2 arrangement. They are just cell membrane folds which are very long, right? So that's about simple epithelium. One.
Yes, ma'am.
And your weekend syllabus is up to area. You can join chemistry.
Angle is 109 degree 28 minutes. So for that kind of angle strain, we need to reduce that angle strain. And that is what we are doing. In order to reduce the angle strain, we are cleaving it. Yes, Pranita, tell me how you would like to cleave it.
Sir, the carbocation should be formed at the tertiary carbon. Sorry, secondary carbon. The methyl group is it.
Okay. So this is OH. This is a carbocation. So this will be what? What I should write here? First of all, tell me this will be OH or OD? Write the steps, everyone. Write the step. Protonate it first, then break it, make the carbocation, attack through D2O, remove D+. You know the steps of the reaction. Huh? The first and foremost is protonation. Protonate the oxygen of oxytane. For the So D plus is having as an acid. You gotta practice it. Look, if you'll not practice, now we have two options to break, either from here or from here. This is more stable carbocation. So it will break from here, we'll get OD, and then we'll get this carbocation. And after this, D2O will break. And you will have OD, plus, and OD. And then after D plus is removed, we get OD. Then we get O. This is what we get. See carefully. Clear, everyone? Mehra,
Yeah, sir, I got it.
Nipuda, are you even getting it or
I
not?
got it, sir.
So now do it for the second. Take NaOD, and D2O and cleave the ring. Now base will attack less hindered site. Okay. Are we good, Arpit?
Yes, sir.
Sir,
Yes.
sir, like in the exam, what is the final answer for that interhalogen anion question?
ICl2 minus.
Sir, but in the final key changes it was not given. So,
Was final key change was given to you.
yes, sir. So posted.
It will be posted again, okay?
Sir,
Yes.
sir, can you like give me the exact definition of interhalogen complexes?
Interhalogen compounds are found by two halogens. Okay, they are just more reactive than as compared to the normal halogen. Okay, one less electronegative, one more electronegative. Like just like take example of ICl. Okay, and moreover, there is nothing like interhalogen anion to exist. So saying interhalogen anion is an absurd thing. You got marks only because there is no other option. Interhalogen cations will be possible, but anions will not be possible.
Sir,
Yes.
sir, in my reference book, there's one line which says that all polyhalide ions are interhalogen ions.
In which reference book?
OP Tandon.
यार ओपी टंडन पे विश्वास नहीं करते हैं यार.
Sir, online also in some research papers, they've said that I3 minus is interhalogen.
Now, look, the problem is that we decide I3 minus as a interhalogen just by looking this, that I3 minus is formed by I2 and with a coordination of like this. Okay. That is why it becomes sp3d and then it goes linear. Just send me the research paper. Okay.
Okay, sir. Yes, sir.
You are having the research paper about it.
Yes, sir.
Okay. You send me the research paper, I will change it. If it is given as a research paper, I will see through it. Because in the normal cases, the interhalogen cations are defined in NCERT. That cations has their existence, and hence we do not have any existence.
Sir,
Mm-hmm.
sir, I have a doubt of the mechanism which you just did.
Mm-hmm.
Beside oxygen, so the bond between the carbon attacked and the oxygen would break, right, sir?
So that is what is breaking. Yeah.
Then the compound would be different, right, sir?
Why it should be different?
Sir, because how come O minus is formed at the place where the tertiary place? If the bond between the attack spot and the oxygen is breaking.
Why O minus will not be formed? O
O minus will be formed over the place where there is O initially, right, sir?
No. You are highly mistaken. OD minus is a nucleophile. And here O minus is a leaving group, and leaving group is only there because it is having a high strain. Now the nucleophile is attacking at this position. Why it's the virtue? Because it cannot attack as a more hindered site. SN2 cannot take place here. So now SN2 attack is here, and this is the leaving group. So we are having O minus as a leaving group. Now this leaving group is protonated again with deuterium. Okay.
No, sir, I didn't get it. I didn't still get it, sir.
Why didn't you get it? You have
So
this. You have this. This will be going to break only because of the strain. Now you have two options: either break from this side or break from this side. This side, it will break from this side only when it is in acidic media. Carbocation can be formed. This carbocation will be stable. But when you are breaking it with a base, OH minus or OD minus, this will break from this side.
Yeah, sir, I understood that part, but O minus is found where it is, right, sir? You drew it at the tertiary carbon side.
So what? What about O minus? O minus is a leaving group, just like Cl minus, Br minus. It just left that carbon. Why we are bothered about it? The OH attacked here, and then at the very moment O minus was formed. That's it. So what is the problem? Why you want OH to attack here? Do you want it?
No, sir.
Then what other option you have? You have two options now. Either you can attack here to break it, or you can attack here to break it. In acidic media, we attack here and we don't attack here. Basically, it breaks by itself and form carbocation. If you are